Welcome to Master Books Biology Lab 3. And in this lab, we're going to need these materials. We're going to need cornstarch, sucrose. Cornstarch, by the way, is glucose. Sucrose, which is table sugar, distilled water, the stoppers from the supply kit for the test tubes, an iodine solution from the supply kit, eyedroppers, a metric ruler. Instead of a wax pencil, I used a Sharpie marker to label things. And then we'll need some food items for the second part of the carbohydrate experiment. So the purpose of our lab experiment is to demonstrate how starches from food sources are broken down by the saliva in the mouth before passing them into the digestive tract. So here's the setup. Here's what I have already done. I have labeled test tubes 1 through 4, and I marked the 3-centimeter line from the bottom to the top of each test tube with my Sharpie marker. Now, as you can see, these are contents of test tubes 1, 2, 3, and 4. In 1 and 2, I put half a ml of cornstarch, which is glucose, and then I added water to the 3-centimeter mark. In test tubes 3 and 4, I put half a milliliter of sugar plus water to the 3-centimeter mark. Now to number 1 and number 3, I added 5 drops of saliva. Saliva contains the amylase, which breaks down carbohydrates. And then to number 2 and number 4, I added 5 drops of water. And those will be our controls because it's just cornstarch and water in number 2. And it's just sugar and water in number 4. Whereas number 1 and number 3 have water, or number 1 has water, cornstarch, and saliva, and number 3 has water, sugar, and saliva. So after we wait the requisite 30 minutes, we'll come back and we will do the iodine test to check for the breakdown of the carbohydrates. So about every 5 or 10 minutes, I'm coming over to these test tubes and I'm shaking them up really well just to make sure that they are mixed and that the proper breakdown that we need to see occurs. And I just wanted to get that on video to show you that it's not necessarily a passive wait time, but we are actually participating in the experiment by just shaking them up and making sure that it's all homogeneous within the test tube. I don't want the cornstarch sitting on the bottom separate from the saliva. You want to have it all nice and mixed up. So every 5 or 10 minutes, come over to your test tubes and shake them up really good, and then you can set them back in the rack. Okay, it's been 30 minutes since the mixtures were formed and since I started the timer. And so now we're going to add 1 drop of iodine solution to each one of these test tubes. All right, I'm going to put these rubber stoppers back into the test tube. And I'm just going to shake them around a little bit just to get them mixed in well. Get all the iodine down into the solution is what I want to have it happen. So looking at each of these tubes, number 1, which has the cornstarch, the water, and the saliva, we can see that there's not a great reaction with the iodine and that combination. In fact, it's quite light in color. So that tells me that the saliva has broken down the carbohydrate that was in test tube 1. Now in test tube 2, we have the same mixture except for the fact that there's no saliva, so there's no amylase to break down the glucose. So that is why the iodine has detected a large amount of glucose and carbohydrate in this test tube, is because the amylase has not broken it down. Now in number 3, there's almost no reaction whatsoever because we had sugar, water, and saliva, and the saliva has broken down the sugar almost entirely, and there's absolutely no reaction with the iodine and the sugar. In number 4, where we just had the water and sugar, you see that there is indeed a slight reaction with the iodine and the sugar, but not a lot, because the sugar itself is not going to be as strong of a reactant, not going to have as strong a reaction as the glucose, which was in the cornstarch. So as you can see, when the saliva actually does help to break down carbohydrates before it enters the digestive tract. And that's what we'll record for each of these. We had a very slight reaction with number 2. We had a very dark reaction with number 3. We had almost no reaction. And with number 4, we had a slight reaction. And those are our results for the first part of experiment number 3 on carbohydrates. So now that we know how iodine reacts with carbohydrates and how it really starts to turn dark in their presence, we're going to test some foods and test their level of carbohydrate. So here I have an apple and corn and a slice of banana and a slice of potato. So I'm going to add 1 drop of iodine to each one of these. Or more. Looks like I might need to add a little bit more just to see what the reaction is going to be. So it will have a reaction because the corn itself was not really big enough with just 1 drop for you to see anything. So let's let that sit for just a few minutes. And see how it reacts. So in looking at the apple, there's not a whole lot of reaction there on the apple. The potato has some darkening here. You can see on the edge right here, the potato, let me use my tweezers. You can see the potato has some darkening there around the edge. The apple really doesn't have a lot at all. Now looking at the banana, you scrape a little bit of this off and see what we get. We get a little bit of a reaction down here, not tons, but the corn, which is what we get cornstarch from, has had major reaction. It's extremely dark. So we can note that of all the reactions, the corn was the strongest, followed by the potato, then the banana, and the apple had the least amount of reaction.